Describe the structure of a blastocyst.
Blastocyst contains outer single layer of trophoblast cells, inner cell mass, which is the source of embryonic stem cells. Hollow fluid-filled cavity called blastocoil.
Describe the consequences of storing the placenta, umbilical cord blood after birth.
It takes a lot of storage space. Storage is expensive sometimes. The precursor cells of conditions like leukemia are already present at birth. Explain how to prevent polysperm EE after fertilization.
The channels in the cell membrane of ovum open and close. Therefore, inside of the egg cell becomes electrically positive than outside. Normally inside is negative and outside is positive.
Cortical reaction. Cortical granules in the cytoplasm of egg cell release enzymes into the zona pellucida. These enzymes destroy the sperm binding sites and hardens the zona pellucida.
The embryo cell and the endosperm cell contain different quantities of DNA. Why?
The female gamete and male gamete are haploid. An embryo cell is formed by the fusion of haploid male and haploid female gametes. The embryo is diploid. One haploid male nucleus fuses with the diploid polar nuclei and form endosperm. Endosperm is triploid.
Explain why the incidence of cystic fibrosis has decreased in recent years.
More increased genetic screening. More carriers are identified. Identification of affected embryos are more reliable. More couple choosing not to have child. More migration. Couple go for IVF, abortion. Change in population diversity. Explain the ethical concerns linked to genetic screening.
Screening may result in abortion. Screening may produce false result. Spare embryos from IVF are destroyed, which is taking human life. An embryo has the right to live. Individuals who are genetically linked may be exposed to unwanted facts.
Explain how histone modification affects gene expression.
Histone modification include acetylation and methylation. In methylation, a methyl group is added to the lysine, which leads to the formation of hetero chromatin. This leads to silencing of gene and the protein is not produced. In acetylation, an acetyl group is added to the lysine. This leads to the formation of EU chromatin. As a result, the gene is expressed and the protein is produced. The development of embryo into either a male or a female is determined by two different proteins. Both these two proteins are the product of the same gene. Explain how this gene can give rise to male, female.
RNA splicing or post-transcriptional modification. Introns are removed and exons are rearranged.
Translation of mature mRNA results in different primary sequence of amino acids or secondary structure of protein. Some proteins result in female-specific cell development and another for male development. Transcription factor produced will activate genes responsible for male-female development. Explain genetic variation. Add a note on its types. Genetic variation happens during sexual reproduction, during meiosis I, due to crossing over and independent assortment. Genetic variation can be of two types. 1. Continuous variation. 2. Discontinuous variation.
Continuous variation. The characteristic is coded by many genes or they follow polygene C inheritance. The environment has an effect on the characteristic. Example: height, weight. Discontinuous variation. The characteristic is coded by a single or a few genes. The environment has no effect on the occurrence of the characteristic. Example: sex, blood groups.
Explain the ways in which the mammalian egg is specialized for its function.
Egg cells are haploid, which ensures the zygote is diploid. Egg cell contains cortical granules to prevent polysperm EE. Egg cell has lipids, fats as source of energy. Egg cells release chemicals to attract sperms. Egg cell has mitochondria which provides ATP.